Hi, welcome. This is group 3. We're going to go over the lesson 12 group problems today. It's going to be a lot of explaining, so this might be a little longer than it usually is, but we'll try and keep it down to those 15 minutes. We're going to start off with Eliza is going to cover number 1.
Alrighty, so starting off with A, which of the terms describes or is an example of a regulatory protein? That would be the activator, repressor, and the CAP. Which of the terms describes or is an example of a small effector molecule? An inducer, allolactose inhibitor, and CAMP. Cyclic AMP. Which of the terms describes which of the terms describes is an example of a sequence found within the DNA? -10, +1, lac A, lac Z, and operator. Which of the above is not involved in the lac operon, the inhibitor? Which of the above is an example of a structural gene? Lac Z and lac A.
Sweet. So we're just going to go on to number 2, which is Sophia.
Okay, so it's just the fill in the blanks, and it's so the lac operon is regulated by 2 inducers, which is allolactose and CAMP. And then the binding site for sigma factor and RNA polymerase is called LAC-P, while the CAP site is the binding site for the activator that regulates the lac operon. And the enzyme adenyl cyclase, how do you say that right? Adenyl?
Adenyl.
Converts ATP into CAMP. The -35, -10, and +1 sequences are found within the LAC-P portion of the lac operon. The LAC-L gene encodes the lac repressor, while the LAC-Y gene encodes lactose permease. And attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And an E. coli strain that contains 2 copies of the lac operon is called a merozygote strain. Number
2. So we're going to go on to number 3.
All right, so I have number 3. So it's about whether, depending if IPTG or glucose and XGAL, if they're absent or present in the cell, if they will cause the XGAL to be metabolized, making the E. coli cell blue. So in the first one, the lac operon will not be expressed because IPTG is absent, and that is similar to allolactose. And so there will be no transcription, and there will be no beta-galactosidase to then metabolize the XGAL. So the cell won't be blue. And then for B, IPTG is present, and glucose is absent, and there is still XGAL present. And because of that, the lac operon will be expressed, and beta-galactosidase will be produced, and that will be able to metabolize the XGAL, making this, causing the cell to become blue. And then for C, it's the same except there's no XGAL present. So because there's no XGAL present, there's nothing for the beta-galactosidase, right? To metabolize, so the cell will remain not blue because XGAL is what causes the cell, when it is metabolized, causes the cell to be blue. Then for D, because glucose is present in this one, lac operon will not be expressed, and there will be no beta-galactosidase to metabolize the XGAL, so also cause the cell not to be blue. And then the last one, even it's basically the same problem. There's glucose present, so the cell will that operon will not be expressed, and no beta-galactosidase will be expressed. But there's also no XGAL in the first place, so even if there was no glucose, there would still not be any. The XGAL will not be metabolized, so it's still remain uncolored or not blue.
Yeah. And then I'm doing number 4, right, Sky?
Yep.
Sounds cool. So I've got number 4. Show my pretty face real quick. So number 4, this is pretty much you're going to be reading the genes themselves, and how you read these genes, it actually gives you a description of the genes as it kind of goes on. And so it says negative refers to a defective gene, so like IL. Lac L refers to a defective Lac L gene. Plus is wild type, and so on and so on and so on. We'll go through these as they come up. So the question asks us to which of these genes is constitutive, inducible, or non-inducible expression of the Lac Z gene. And so the Lac Z gene is here. And so this A is wild type, so it's going to be inducible. That's a structural gene there, so it's going to work. And then as we go down, we have the Lac S, or the Lac L gene has an S on it, which this produces a Lac repressor that cannot bind to the operator, or can bind to the operator but cannot bind to the L-lactose. This means that the repressor will not leave the operon, or it can't unbind. So the repressor is pretty much stuck on there, and so it can't leave. And that's going to make it non-inducible. Doesn't matter what happens, it's not leaving. And then as we go down, number C, we now they throw in a plasmid in here, and so we have Lac L's good, but then it has this P and the C. So the P is really not going to do much because we have a wild type P on the plasmid, so it's going to work either way. But the kicker here is the OC, and the OC refers to a defective operator sequence. The Lac repressor cannot bind to the OC, or the O, Lac O, and this means it's always going to be running. It's always going to be running, always going to be on, and so it's going to be constitutive. That's why it's always going to be on. As we go on, the next one, it's asking, we have a wild type chromosome here, and then it has an IS, a P, a O, and a wild type Z. So this IS on the plasmid is going to be just like B. So the first thing's going to happen, it's going to hit that IS, and it's going to produce that repressor that cannot unbind. And so it's going to unbind the plasmid as well as the chromosome. It's going to go to both those because they're both in the cell. That's going to be non-inducible, just like B is. And the last one, so yes, it produces a repressor that cannot unbind, but it also produces a OC, which your repressor cannot bind to, so it's going to be constitutive because the plasmid is always going to be producing that. So that's kind of how, I hope I explained that well enough for number 3, or number 4.
Okay, then number 5 is basically just asking you, you end up doing a lot of the same stuff you do as in number 4, but you're adding lactose or not in the test tube, and you have to tell how much yellow is made. 0 meaning no yellow, 1 is kind of like the middle amount of yellow. It says indicates the amount of yellow product when 1 copy of Lac operon is transcribed, and 2 is when the 2 copies of the Lac operon are just transcribed makes the most yellow. So basically you end up doing the same thing as to what Roy was describing in reading the genes in number 4. And so for A, when you have lactose or when you don't have lactose, you get no yellow color. So it's 0s for both because it's non-inducible. For letter B, you get 1 with lactose and 1 without lactose. C, you end up getting 2 with lactose and 1 without lactose. D is 1 with lactose and none without lactose. And E is also non-inducible, so you get 0 with both of them. So without going through all the major steps of that, it's basically the same as number 4. But, just adding lactose and figuring out how much yellow, so.
Yep, that's kind of how it works. I hope our explanations helped out a lot, and have a good night.